Molecular genetics and inheritance. When you say molecular genetics, it is about DNA, gene, chromosomes, and cell division. These are at molecular level or not at phenotypic level. It is at molecular level. DNA, what is DNA? The abbreviation DNA stands for deoxyribonucleic acid, and this is the molecule that holds the genetic information for a cell and an organism. You can consider it carries the information from generation to generation. You know what is information. Okay? A DNA molecule contains a code that can be used by a cell to express certain genes. Specific sections of a DNA molecule provide the information to build specific proteins. This specific section we call gene. This process of converting the specific gene sequence into a specific protein is called gene expression. So this is the code in DNA expresses into the protein. DNA comes in the form of a long linear molecule referred to as a strand. We call DNA strand. Each strand of DNA is bonded to a second strand of DNA to form a DNA double helix. In eukaryotic cells, DNA is found in the nucleus as a tightly coiled double helix. We will see the DNA structure and the helical structure and the like. It says in eukaryotic cells, it found in the nucleotide. It found in the nucleus. Because what about for prokaryotes? Because prokaryotes do not have membrane-bound nucleus. Therefore, the DNA found in prokaryotes in the cytoplasm by floating in the cytoplasm. DNA molecules are replicated during cell division. This is, you have to note that in each and every cell division, there should be the division of genetic material. DNA is one of the two genetic materials. What are the two genetic materials? DNA and RNA, the two acids, deoxyribonucleic acid and ribonucleic acid. So, in each and every cell division, there should be division of or replication of the genetic material. When a cell divides, the two new cells contain all the same DNA that the original cell had. In sexual reproduction, with two parents, half of the DNA of the offspring is provided by each of the parents. Offspring means child or baby. So the genetic material or the DNA of a baby is from the two parents, father and mother. Half is from mother and half or 50% is from father. For example, let's say this couple are the parents for some children. During gamete formation, from the male, the chromosome will be the X and Y chromosome, which are the sex chromosomes. And from the female, X and X are the chromosome for gamete to be distributed to gamete. So during fertilization, if the gamete of X chromosome come from male and X from the female, then it will be XX and the individual will be female. If the gamete carries Y chromosome from the male parent and if randomly X come and joined with Y from the male during fertilization, then the individual will be male. The same is true in this case. If X from male and X from female, XX, then the individual will be female. If Y come from male individual and X from female, then XY, the individual will be male. The ratio is 1 to 1 because XX female, XX female, 2 female, and XY male and XY male, 2 male individuals. So 2 to 2 to 2 ratio means 1 to 1. The condition is, or the probability is equal. Nature does not bias. The structure of DNA and chromosome. Chromosome is a thread-like structure that is made up of DNA and histone. This is a kind of protein, histone protein. DNA is a molecule that stores genetic information, whereas histone is the core of a chromosome around which chromosomes DNA wrapped. These histones are the tertiary kind of proteins. There are 4 levels of protein structure: primary, secondary, tertiary, and quaternary. The histones are the tertiary kind of proteins. Most of the proteins, functional proteins, are the tertiary kind. The loosely organized form of chromosome is throughout the nucleus in loops. When the cell is not dividing, is called chromatin. If the cells are in division state, then the chromosome, we call that chromosome because it will be condensed and visible. But without cell division time, we call the things chromatin. Individual chromosomes are not easily distinguished unless condensed. Unless it become condensed and thick, then it will not be visible. Visible by microscope, not by eye. You can consider it is even not visible by microscope. It is very thin and a small entity. On the condensed organization of chromosome, genes are inactive, while uncondensed or loose organization allows the genes to be active. Genes are active during chromatin stage and inactive during chromosome stage. As a cell prepares for cell division, the chromatin loops will have duplicated themselves and become compacted or condensed to form a chromosome, which is visible under light microscope. It will be visible only during cell division. This is a general chromosome structure at metaphase stage. There are different phases in cell division. One stage is, or the middle stage is metaphase stage. During that, the chromosome became visible. At that time, you can see centromere and the constricted region, and also this one thread is, or one strand is called chromatid, and the other is also the other chromatid. They are called sister chromatid, and they together called chromosome. This is a fiber-like structure we call chromatin, which is made up of DNA and proteins. This is a simple structure of chromosome. This is a chromosome structure. The same thing. For example, this is a DNA, the helical structure. Then these bead-like structures are histone proteins. And if the DNA or this chromatin fiber wrapped around the histone's protein, we call nucleosome. And finally, if this chromatin structure will be condensed, then we will see the chromosome. DNA is made up of 2 strands of polynucleotides. There will be many nucleotides come together because nucleotides are the monomer units of the DNA. So the 2 strands of DNA is made up of the polynucleotides joined together and twisted into a double helix, and the strands are anti-parallel to each other. So the 2 strands are anti-parallel. This is the structure of DNA. The basic unit of DNA strand is nucleotide. That is, the building blocks of a DNA molecule are the nucleotides. That are the monomers of DNA are nucleotides. There are 4 types of nucleotides: adenine containing, guanine containing, cytosine containing, and thymine containing. In DNA, thymine; in RNA, it will be uracil. All nucleotides have a phosphate group. The 3 components in one nucleotide or in a nucleotide are phosphate group, pentose sugar or 5 carbon sugar, and the nitrogenous bases, one of the nitrogenous base per nucleotide. The nitrogenous bases in nucleotide are adenine, cytosine, guanine, thymine or uracil. Thymine in DNA, uracil in RNA. There is no thymine in RNA, there is no uracil in DNA. The structures of deoxyribose and ribose in DNA. What is the difference between deoxyribose and ribose? Is very simple or very slight difference, which makes them that much or huge difference. That is at carbon number 1, this 1 prime is for this carbon, so carbon number 1, carbon number 2, 3, 4, 5. As this carbon, the second carbon, there is no OH, only hydrogen here. There is no hydroxyl group here in the deoxyribose sugar. In ribose sugar, as the second carbon, here is OH. That is one difference. The only difference is at the second carbon position, where RNA has hydroxyl group and DNA has only hydrogen. This is a nucleotide. A nucleotide has 3 components: this phosphate, the ribose sugar or pentose sugar, that is 5 carbon sugar, and the nitrogenous base, either adenine, guanine, cytosine, or thymine. Notice that the 1 prime, 2 prime refers to a numbering system for the carbon atoms that make up the sugar. Simply you shouldn't write 1, 2, 3, 4, but use prime, 1 prime, 2 prime, 3 prime for numbering the carbon. This is to differentiate other molecules within the molecule. There are hydrogen, there are oxygen, and other elements. Therefore, to identify the carbon, which is very important and the backbone element of the organic molecules, is considered in that intention, and the numbering is in this form, 1 prime, 2 prime, and etc. Nitrogenous bases. There are 2 major nitrogenous bases: Purine bases and Pyrimidines bases. The Purine bases are adenine and guanine, and the Purine bases has double carbon nitrogen rings. There are carbon and there are also nitrogen. That is why they are called nitrogenous bases. And the rings are 2: one ring, one circle, the other ring. And pyrimidines have single rings of carbon, nitrogen. Thymine and cytosine are the pyrimidine bases. This is only one circle or one ring. Bonds between the sugar in one nucleotide and the phosphate group in the next hold the nucleotides together. When nucleotides come together and make the DNA molecule, as we said, the nucleotides are the building blocks of the DNA molecule. How they make bond is the sugar of one nucleotide bond with the phosphate of the other nucleotide. The base does not have a role in this case. Okay, it does not take in this linking of the nucleotides in a strand. In one strand, there is no involvement of bases in bond formation, but it will have role when the 2 strands come together, as we will see now. This is why we call sugar phosphate backbone. The strand is called sugar phosphate backbone. The nucleotides in one strand are paired with the nucleotides in the other strand according to the base pairing rule. That is, here you can see there will be involvement of bases in the bonds formation between the 2 strands to form the double strand. Adenine always pair with thymine. If it is RNA, it will be uracil. Cytosine always pair with guanine. DNA is very stable molecule at normal temperature. The hydrogen bonds hold the 2 strands together in position through the bases. how the DNA is stable is because of this. Okay, the hydrogen bond between the two strands make the DNA will be stable than compared with the RNA. The stability of the DNA molecule is important because it ensures the genetic code in the DNA molecule, which does not become corrupted. Okay, there will no easily there is no error formation during DNA replication. This is because of these features. Base pairing rule, that is, purines only pair with pyrimidines. Purine will not pair with purine, or pyrimidine will not pair with pyrimidine in the normal condition. Or purine always pair with pyrimidine. That we call complementary base pairing. For example, adenine pair with thymine, A with T. Cytosine pair with guanine, C with G. We call this Chargaff's rule. C with G, T with A. 3 hydrogen bond is required to bond guanine and cytosine. We said always cytosine with guanine, there will be how many hydrogen bonds? 3. 1 hydrogen bond, 2 and 3. And 2 hydrogen bonds are required to bond adenine and thymine. We said A always pair with T or thymine. In between, there will be 2 hydrogen bonds. That is why adenine always pair with thymine and guanine always pair with adenine with thymine and guanine with cytosine. This is their structural conformation, which is make this rule, okay, the base pairing rule. What is just the DNA molecule or the DNA structure look like? We said this is considered one strand, this one is the other strand or the backbone. The 2 backbones are these. In the backbone formation, we said phosphate pair with the sugar. Here is phosphate, then here is the sugar, then here is what? The nitrogenous base. Then phosphate, sugar, phosphate, sugar, phosphate, sugar, the backbone. In this case, phosphate, sugar, phosphate, sugar, phosphate, sugar, or sugar, phosphate, sugar, phosphate, and like that. But the bond is between what? The nucleotides. This is one nucleotide. Phosphate, sugar. Here is nitrogenous base, one nucleotide. Phosphate, sugar, nitrogenous base, then one nucleotide. And then phosphate, sugar, we expect here nitrogenous base, one nucleotide. Then it will continue in such a way. But in the backbone formation, there is no bond between the bases with other molecules. But after formation of each strand, and then when the 2 strands come to form linkage, there will be involvement of the nitrogenous bases from the 2 strands. From this strand, here is thymine. From this strand, here is adenine. Then thymine with adenine. From this strand, then cytosine. From this one, guanine. Then guanine, cytosine. Then consider this kind of bond formation for hundreds, thousands, millions, and billions of nucleotide bonding. In this case, you can see 5 prime. This is the 5 carbon from the pentose sugar, 5 prime to 3 prime. This runs from this to this. And this one from 5 prime from this direction to this direction. They are anti-parallel. More illustration. Here, this is a double helix structure of DNA. And here you can see one strand is moving from this direction to this one, from 5 prime to 3 prime. And the other strand from this direction to this direction, from again 5 prime as this end to 3 prime to this end. Generally, the direction of the DNA is from 5 prime to 3 prime. That is because when DNA replicates, the nucleotides will come at the 3 prime end of the growing strand of the DNA. It will not be added to the 5 prime. It will be added to the 3 prime. And that makes the DNA structure will be anti-parallel. One from 5 prime to 3 prime to one direction, the other is to the opposite direction. Here you can see the bond between the two strands is the nitrogenous bases, adenine with thymine, guanine with cytosine, and the like. The backbone is here phosphate bonded with the sugar, then again phosphate with sugar, phosphate with sugar. This one is one nucleotide, phosphate, sugar, nitrogenous base, one nucleotide, phosphate, sugar, nitrogenous base, the other nucleotide. So the bond between this nucleotide and this nucleotide is between for this phosphate and this sugar. Thanks for watching.